Welcome everyone. I assume many of you are doing a sort of like a pub crawl, a virtual open day, so welcome to this one. For your information, this is being recorded and will be available on web pages that I believe you can see in the chat part of the presentation. Also want to alert you that the projects are available via a link also, which I believe you can see via the chat section of the Zoom presentation. My name is Melissa Southey. I've got the pleasure of hosting next 30 minutes to take you through some of the projects that are being offered for honours and postgraduate research interested students in the Department of Medicine in the Monash Health Translational Precinct. This is going to be focused on cancer and cancer research theme, and welcome for those of you who are interested. Some of my colleagues will be able to address questions as you have them, so please enter them into the question section of the Zoom, and we'll try, and if we don't get to them during the presentations, we'll come back to them at the end, presuming that we have time. So as I said, we're the Department of Medicine, and the Department of Medicine is a very diverse department. For those of you who heard Eric Moran present the school in a session about an hour ago, he would have covered some of these elements of the Department of Medicine. But we are a very diverse department located in the Monash Medical Centre with a large number of interests and diverse research activities embedded within clinical and also basic research activities across the precinct. In this session, we're really going to be focusing on these three parts of the Department of Medicine: cancer, precision medicine, and also some aspects of supportive and palliative care. I really recommend going to this part of the web if you would like more interest, if you would like more information in projects that you're interested in. You can click through any of these themes in the department and learn a lot more about research that's active within the department, projects that are coming up relevant to students, and other activities like seminars and other media type engagements that you might be interested in joining. We hope that it won't be too much longer before we can do some of these things face to face rather than via these rather strange media interactions with you. But anyway, this is the department, and this is the focus of the presentation this evening. So we're going to have a number of presentations. We're going to have some presentations from precision medicine. Then Eva Segelov is going to talk specifically to opportunities in oncology. And Poon, I hope, is going to be able to present some projects available in the supportive and palliative care aspects of the department's work. We also have a PhD student who has freshly joined us from submitting her thesis, who will be able to answer more questions about the student experience within the department, who is online and will be able to answer your questions in the chat box as well. So first to precision medicine. What is precision medicine? And this is a really diversely defined discipline. In fact, it's a bit cheeky probably to call an activity precision medicine, because of course I think everyone in medical research and clinical medicine aims to improve the precision of their practice. But in very broad terms, it really is about the creation of new evidence, and that is across the landscape of medicine. We have activities in the Department of Medicine that really are involved in every one of these terms that I've used in the top blue box. Cancer prevention, for instance, screening and diagnosis of disease treatments and so forth. It even includes bioinformatics and some analyses of the cost effectiveness of delivering medicine in a large number of aspects that we're all involved in our day-to-day work. And of course, the creation of new evidence really is to improve health outcomes. So precision medicine is about targeting health interventions, saving money, and better understanding the diversity of disease and the diversity of humans in a very general and sometimes very specific sense. So I'd sort of like quoting John Maddick here. Precision medicine is really about moving from the art of crisis management into a science of good health. So, right. Welcome, Peter. Nice to have you. So one of the interests in precision medicine is that we have a very expanding interest in DNA methylation as a biomarker for disease. One of the activities of the precision medicine team has been to be very attentive to the collection of biological material from studies over the last number of decades. So we have a very well-established biorepository with a very established and large series of biological specimens collected from humans in all sorts of disease contexts across their lifespan that we can now use to address questions that are relevant to precision medicine today. And one of these attributes is DNA methylation. And we have several large systems in place to examine DNA methylation. And I'm showing you one in this slide. This is a system that's been produced by Illumina. We use EPIC arrays that measure methylation across the genome at 800,000 sites across the genome, and then use bioinformatic approaches to examine the data generated from the array to look at changes in methylation in disease states and try and identify really good biomarkers for disease. We've been using this for some time in the context of identifying markers that identify people at increased risk of disease. We're also moving on to using these markers actually in the disease context to be able to identify people with particularly aggressive disease and people that might be more responsive to certain therapies rather than others. So this is a very large initiative in the team, and one that would be available to students interested in the projects that are coming in subsequent slides. So this is another system that we have in place. This is a Biomark from a different commercial producer called QIAGEN. It has similarly able to use our very large biological resources. The data output is a bit simpler, but again is very precisely able to measure DNA methylation across our biological collections, which include specimens of DNA and also specimens from material dissected from or surgically removed from patients with, for instance, cancer specimens. So to talk to one project more specifically, we're interested in looking at DNA methylation as a biomarker for aggressive prostate cancer. In particular, we're interested in looking at a molecule called the Volt RNA21. This is quite a remarkable molecule. I don't probably have time tonight to dwell on the detail too much, but it's only 100 nucleotides long. It's a non-coding RNA, but methylation of the DNA is extremely important to the regulation, and it's known to be a tumor suppressor gene. So the hypothesis of our study we're proposing is that the assessment of DNA methylation, including methylation at the Volt RNA in prostate cancer tissue, will identify pathways involved in aggressive prostate cancer. So the aims are to use the EPIC array, which I showed you in one of the previous slides, to measure genome-wide DNA methylation in prostate cancer tissue, to confirm the DNA methylation measured using the array, using the alternate methodology, using the Pyromark assay, and then to use the data acquired in both of those methodologies to identify pathways involved in prostate cancer that are associated with Volt RNA methylation status. We've been particularly excited by recent data from the laboratory where we think that the methylation of the Volt RNA is truly associated with aggressive prostate cancer, which is an extremely important part of prostate cancer spectrum to identify new biomarkers for and hopefully identify new cancer treatments that are targeting the methylation in this region. This project is likely to involve both wet laboratory work and dry laboratory work. There's a lot of upfront detail in molecular pathology approaches and also some bioinformatics to analyse the data as it comes out of the instruments to try and approach this part 3 of the project aims. So at this point I will hand over to Pierre. Dugue, if you could come online. I think it's just a matter of unmuting yourself.
Yes, I'm here.
Thank you, Pierre.
Thanks, Melissa. So I'll talk a little bit about the research I've been doing over the past few years, which is around DNA methylation and cancer risk. So not lab work. I mean, of course, working with the lab to generate the data, but most of my research is about data analysis and genetic epidemiology as well as bioinformatics method. And there are a number of projects we can do with this data. So as Melissa explained, DNA methylation is one epigenetic mechanism. And as you can see on that beautiful picture, it's the addition of methyl groups along the DNA, which guides the expression of the genes. So if you like the DNA, it's the notes on the music, and the methylation, it guides the expression. And it's influenced by the underlying genetic sequence, but also with a number of factors over the lifetime, such as aging, which is a strong determinant of DNA methylation, and that's actually why we lose physiological integrity, but also many lifestyle and environmental exposures. So it's quite dynamic, and those lifestyle, aging, and environmental exposures, they cause cancer. So it's very interesting to study in this context. Most of the analysis I did was using a microassay targeting almost 500,000 sites across the genome in the blood of around 7,000 participants. So that's a huge amount of data. And with new technology, we can measure even more, like 100,000 CpGs or even the whole genome. It's also, most of the research was using blood samples, but in fact, any tissue can be profiled for DNA methylation. One big line of research was to conduct epigenome-wide association studies. So you might have heard about genome-wide association studies. So that's the same thing with DNA methylation, where we tested at each of those sites along the genome the association between risk factors for cancer and DNA methylation. And we also investigated which biological pathways were involved and how the methylation changes with factors such as smoking cessation, for example, with the goal to discover and of course replicate using external data, objective and robust biomarkers of exposure to risk and cancer risk. So the next step after identifying those markers was to test whether these methylation marks were associated with cancer risk and also cancer survival, cancer mortality. A lot of what I did was to use cancer-free blood from cancer-free participants and assess their risk of cancer over many years of follow-up. And using those data, we studied a number of methylation patterns in the blood, such as so-called epigenetic aging, or the global hypomethylation pattern, or methylation marks of smoking, alcohol, obesity, as I explained, and even more recently, maternal smoking, because it's actually possible to track in the blood of an adult whether her mother smoked during pregnancy, so a long time before. And our goals are to gain a better understanding of the causes of cancer and improve our ability to predict cancer risk and the cancer survival. So this is just an example of the studies I'm doing, but there are many more things we can do, and some are already available on the website. Thank you.
Thank you very much, Pierre. Now, hopefully we can just keep turning on. Yes, so I'd now like to introduce Tu. Tu, would you like to come online and talk to you? Welcome.
Thank you, Melissa. Good evening, everyone. My name is Tu, and I work in the same lab as Pierre in precision medicine. I'm a molecular biologist, genomicist, and bioinformatician. My research combines both my interests, genomics and bioinformatics, and focuses on the underlying causes of genetic predisposition to cancer, in particular prostate cancer, pediatric cancer, and breast cancer. You might have heard of next-generation sequencing. The amounts of raw data produced are phenomenal, and many computational steps are required to translate this output into high-quality variant calls. And I'm really excited by the way that technology has evolved and giving me the chance to use my combination of skill sets to make sense of this data. You can use next-generation sequencing to identify genes that are associated with an increased risk of developing a disease. You can use NGS to cost-effectively and accurately assess the prevalence of genetic variants in various populations. And with tumor sequencing, you can also characterize the effect of mutations found in a patient's tumor and identify potential therapeutic targets or clinical trials your patient might be eligible for. So a lot of things can be done, and translational bioinformatics can help us take this large amount of genetic information through to the translational level and make an impact on clinical practice to improve health outcomes. So the project that I want to talk to you about is just one of the many things we work on, but tonight I'd like to talk about this specific one. The project combines genomics and bioinformatics, looking more specifically at one type of genetic variant, those that affect normal splicing of the DNA into RNA, and one specific gene, ATM. ATM is widely accepted as a breast cancer susceptibility gene, but only one ATM variant is currently used in clinical genetics practice in Australia. So we need to generate more evidence at the bench and deliver this evidence to the clinic. So the hypothesis here is that the assessment of spliceogenicity-based probability of pathogenicity can help prioritize variants for further molecular characterization. And the aims of the project are to review, then apply bioinformatics tools for splice site prediction, then identify from a database of ATM variants which one should be followed up in vitro. So depending on the skills and interests of the student, the project could involve a mix of wet and dry lab work, or be 100% computationally focused. That's all I had. Thank you.
Thank you too. And now we're going to hear from Professor Eva Segelov, who is Director of Oncology and Professor of Oncology. Eva, we're still staying within the circle as indicated in our Department of Medicine. Eva, shall I stop sharing screens with?
Yeah, sure. Maybe.
There we go.
If you can see me. So I don't have slides because we cover a large amount of topics. So we are, our laboratory is made up of clinicians, so we're all cancer clinicians. In the beautiful MHTP building, we have a state-of-the-art clinical trials centre down on level 3, just above the cafe. And so it gives us a fantastic chance to integrate and go directly from the lab to the clinical space. So we have a variety of projects and we're always happy to customise projects. We really want to focus on using patient material and patient information to inform the lab. So going back from bedside to bench rather than the usual bench to bedside, but it is a loop. We work closely with Josh Uwe, who you may have heard of in the other session, looking at immunology. Cancer and immunology are irrestricably linked. He's trying to dampen down the immune system in his particular projects looking at T-cell therapy, and we're trying to actually do the opposite and ramp up the immune system to recognise and fight cancer cells in the body. So we've got some projects in that space. The main space we operate in is using clinical trial material to try and find biomarkers for therapies or predictive or prognostic markers. So we have access to blood and tumour samples from large international clinical trials, and we're looking at projects for a particular example, I'm one of the lead investigators of a global study looking at aspirin use versus placebo in patients who've had bowel cancer. And we've collected a whole lot of specimens over time, and we're going to see if we can identify markers for which patients have the cancer return and which patients benefited from aspirin. But as a spin-off, we've now got a large data set of patients, cancer survivors, and we have put in a grant for a study of cardiac health in these survivors. And again, these are all people who weren't on aspirin at the start. So it gives you a good chance to look at this population. So we're very happy to talk to people who want a mix of wet lab or dry lab and clinical experiences. You get the opportunities to write papers with us, usually clinical type reviews, as well as whatever data you generate. And so welcome.
Thanks, Eva. It's going to be hard to decide what to do after that repertoire. But I think you make a really good point. I think many supervisors are really willing to shape a project to a student's interest. So it really is worth speaking to supervisors who you know work in an area of your interest and don't just rely on the published projects. Often the published projects are intended to give a taste rather than to be an absolute locked-in project. So it is always worth contacting a supervisor to have a discussion about a project. Right, so now I can pass the unmute baton, so to speak, over to Peter. Peter, you're going to share slides with us? Or?
Yes, I am.
Excellent.
I must apologise for interrupting you before, because
no problem. It was nice to hear you arrive. Thank you.
I got a message from my PA saying it's your turn, so yeah, I quickly logged on. I think I will share some slides as well, if I can find them. There they are. Right. Is my screen coming up? Share screen?
Yes.
Great, terrific. So along the same theme as Eva, I thought we'd just give you some broad brushstrokes around our supportive and palliative care unit and the research opportunities within the unit. Firstly, just to give you a bit of background, because many of you probably don't know much about us. We're an academic teaching unit, also for Monash University, as well as the accredited training site for the College of Physicians as far as palliative medicine is concerned. And we're one of the 3 recognised sites in Australia for integrated palliative oncology with ESMO, the European Society of Medical Oncology. We have inpatient beds at Monash, McCulloch House at Monash Clayton, as well as inpatient beds at Casey, outpatient clinics, a very impactful consultation service across all campuses. And our patient cohorts include cancer and about 65% and 35% chronic disease patients. The unit has a very strong focus on translational research and very, like Eva, very broad, but I guess generally speaking, it's either very clinical research, such as pain or refractory symptoms, or translational service research, health service research. The team is diverse. Our physicians are broadly trained in palliative medicine. We have dual trained physicians, including in medical oncology, general medicine, and a special interest in digital health. Thus far, we've taken PhD candidates as well as BMed science candidates. And we have a research assistant and uniquely integrated into our clinical team. And we have a very strong team ethos, which is typical of palliative medicine. Indeed, we have 2 multidisciplinary meetings every week with all staff there. So if students are with us, they attend those as well. So not only do they get a flavour of whatever research they're doing, but also they get a better understanding of what palliative care clinical work involves. Some examples of our projects are listed in the handbook, and you can see there's diversity there from very clinical issues like managing delirium, whether there's refractory pain issues, looking at that. But then again, substance lymphedema drainage there as well. But then there's other areas of research looking at telemedicine, telehealth, so digital health in other words. And finally, looking at translational health service research, whether that be in chronic disease, in cancer management. So very broad. And I just put up, I think, 2 examples there. I won't go into depth, but very different types of research. And on top, and in addition to this, as far as our translational research, we really look at the outcomes, very practical outcomes of our service interventions. So we hope that whatever we do has meaning. So our research that is around health services, that we can immediately translate that into a working model, for example, telehealth. So we completed a study over 3 years in telehealth, and immediately were able to utilize some of that during this COVID crisis in managing our own patients. And currently we also have a study involving the use of ketamine in refractory pain, particularly in mucositis pain in cancer treatment. So again, if that is proven to be beneficial, we'll be able to utilize that within our clinical service pretty much immediately. So I welcome anyone's interest. I've sort of given you a broad brushstrokes there. So please, if you have an area that you're interested in, whether it be clinical, whether it be health service related, just contact me and I'm more than happy to have a chat with you. Thank you.
Thank you very much, Peter. I think we just have just a few minutes for questions, and I know many of you will be wanting to move on to the next session. One question's come through, do we offer summer placements and internships? I think it would be universal that we would all do that. It would just be a matter of contacting us and discussing a suitable project. In fact, I think many of us will recommend it for a student considering engaging in postgraduate research, that a summer placement or an internship is a really good taste for what's to come and very worthwhile if it can be arranged.